Okay, so this is going to be part 2 of our oncolytic therapies and gene therapies. So this example in your textbook, they're talking about using a modified poliovirus to treat a brain tumor. So this is a pretty cool example, and there are a number of oncolytic viruses that are in the clinical stage right now or are approved, and you can go to the government website and you can see right now what clinical trials are currently ongoing. Alright, so here we are at clinicaltrials.gov. You can search in the whatever terms you want. So we're searching for oncolytic here. And here we got some search results. So we've got studies that are recruiting, studies that have been withdrawn, studies that have been completed. And you can see right now we've got 156 studies. So recruiting now adenovirus. This is a common virus used in therapy. It's a DNA virus. Adenovirus is associated with usually the common cold or inflamed adenoids, usually very mild illness, and then if you use it for gene therapy, or for an oncolytic therapy, you usually modify the virus. You might take out the pathogenic genes. You're going to target it specifically to the cancer you're treating, in this case a glioma, and then you might engineer the virus to either kill the cancer cells, and you're going to target it again with those specific ways, right? So you can either target the receptor or the transcription or what have you. So we've got here's one for a glioma, looking for solid tumors. You can go through, this one was completed, so a safety and efficacy trial. And we got some more. Here's a recruiting one for colorectal cancer. Colorectal cancer. So if you were interested in any of these, you could follow up. Also of note, you can go to the same website to look for gene therapy trials. So gene therapy is where you're trying to replace a defective gene in a person. So the idea is to introduce a therapeutic gene or a quote-unquote good gene into a person. Excuse me. And you have a lot of the same challenges that you do with oncolytic therapy, but it can be a little bit more complicated. So first, you've got to genetically engineer the virus. You've got to remove the pathogen-causing genes. So you've got to remove the pathogenicity. And then you've got to add your therapeutic gene or your gene of interest. All right, and then you've got to be able to produce enough virus to be effective. And this is not a trivial task. If you are introducing a virus into a person, and remember the liver is going to get almost all of it, then you might have to produce a lot of virus for one person. So there's some cost effectiveness things here that need to be taken care of. You've got to think about what your delivery is going to be. Is it going to be in a patient, or is it going to be ex vivo? I'll talk a little bit more about that. You've got to think about targeting. Now, the gold standard, what everybody's looking for, is you can administer this IV and it goes straight to where you need it to be. Or are you going to, try to inject it into the site of interest? In order to target it, right, you really need to target it to a specific cellular receptor. So if you've got lots of cells in the body, you really need to pick a receptor that's going to be unique. So if you want to target hepatocytes, you're going to use a targeting ligand on your virus that is specific to that receptor. Say if it's hepatocytes, something specific to hepatocytes. Or if you want to target the kidney or the pancreas or whatever it is, you're usually going to take your virus, and you've already engineered the genes in the inside, so you've got your therapeutic gene there instead of the pathogen-causing genes. And then you're going to modify these spike proteins or attachment proteins, so you're only going to get this virus into the cells that you want it to go. The next thing that you really have to think about is trafficking. So assuming you've targeted the right cells, it's going to bind and enter to the right cells. How is it going to traffic through the cell? So we've got our cell here, and we've got the receptor on the surface, and we've got our nucleus here. You've got to be able to negotiate the endosome, so probably going to come in through endocytosis. You've got to be able to escape the endosome. You've got to be able to use the cytoskeleton. That didn't write well at all. You've got to be able to use the cytoskeleton. You've got to be able to get to the nucleus. Remember, the nucleus has these nuclear pores, so you've got to be able to get through the nuclear pore. Then you've got to get into the nucleus. So inside the nucleus, you've got to make sure that you have some control sequences, regulatory sequences, so that your genes are actually going to get expressed instead of just degraded. And so then with that, you've got to think about stability. Do you want this to be temporary? In which case, you might want your gene of interest to remain as an episome. An episome is an extra chromosomal element that is not, so it's not part of your genome. It's like a plasmid almost. And it eventually gets degraded and broken down. Or do you want to integrate? If you integrate, this could be a really long-term solution, but you have to make sure you don't cause cancer when you integrate. Right? And then on top of this, right, you need to make sure that you have good expression. So you want to make sure that your gene of interest is expressed just the right amount, the Goldilocks amount. Not you need enough, right? So not too little that it's not helpful, but you don't want too much, because too much of anything in a cell can be really, really bad. So you want just the right amount of expression. And you need to make sure that the gene product you're producing is translated appropriately and it's modified properly in the ER and the Golgi, and that you have a nice functional protein. Now, if this is going to be a protein that's going to be expressed inside the cell, that's a little bit easier. If this is a protein that's going to be excreted, you have to make sure that it traffics properly, that it is excreted, and it's excreted as a functional protein. And through all of this, you have to make sure that you don't trigger an immune response. So I don't know why my trackpad is starting to get flaky right now, but try to bear with me. That didn't help at all. Oh, not trackpad, tablet. Anyway, you have to make sure that you don't trigger an immune response. So you have to make sure that the virus or the gene that you're expressing doesn't trigger an immune response, because you don't want your immune system to kill the cells that you're trying to save. So there's a lot of challenges with gene therapy. Now, what diseases are good targets for gene therapy? So the best targets are ones that involve just one gene, one broken or defective gene. Then it's a lot easier to bring in just one functional gene, replace one faulty gene. So something like cystic fibrosis is a really nice target, because we know that it's a mutation in just the cystic fibrosis transporter protein, so the one that regulates the ions across the membrane, right? So this is a really nice target. We've got other diseases or other conditions like autism, which is a very complex disease, and many genes can contribute, as well as the environment. So that would be a bad target for gene therapy. So anything that's you can fix with one specific gene, or sometimes using a gene therapy in combination with a medicine can work. But again, we're looking for simple conditions. Now, we're looking for something that we can tweak in the cell relatively easily. So what are common genes of interest? So specific antigens, this is listed for immune modulation. So this might be trying to treat, say, rheumatoid arthritis, or trying to regulate the immune system. Regulating the cell cycle. Regulating sensitivity to drugs. You might deliver gene therapy to make drug treatment more beneficial. Fixing a defective gene, so this would be an example of cystic fibrosis, or proteins that induce apoptosis. So, for example, there's no reason that you can't use gene therapy on a cancer. So you can introduce a virus that is going to trigger apoptosis. And you can go to the that clinicaltrials.gov, and you can see current gene therapies that are in clinical trials today. Now, there have been some big successes in gene therapy. There's also been some setbacks. So here is a situation, and then the first one in 1999, where there were some issues where people, will say the scientists didn't necessarily follow all of the steps that they were going to, and they administered too high a dose of the viral vector, and he had an overly strong immune response, and so he died after a too high dose injection of the vector due to an immune response. Essentially went into shock. Now, after this, there were a lot of reviews over what protocols need to be followed during gene therapy trials, and some improvements to the process were put in place after that. Now, here we've got, in 2003, we have SCIDS patients. So these are patients that are severely immunocompromised. And these patients typically die within the first year or so of their life. And so if your child is diagnosed with this disease, they are going to die. And so this is one of the reasons it was one of the early gene therapy trials, because if there was a problem with the gene therapy, your kid was going to die anyway. And that sounds kind of harsh, but it's easier to get experimental treatments approved if the patient has a fatal disease. You're not going to try out a brand new gene therapy, with untested things for, acne, because it's acne. Even severe cases of acne wouldn't really warrant, extreme procedures. So this trial actually worked initially. And all of the kids, pretty much, that they used the therapy on, they were cured of their immunodeficiency. And so this was a great success at first, and everybody was really, really happy. And then years later, so these kids got several more years of life. Everybody thought they were cured, and then years later they developed leukemia, and some of them died from leukemia. And so it was really, really sad, because you have a baby and you're told your baby's going to die within a year, and there's nothing that can be done, and then you're like, but there's an experimental gene therapy trial that might save them, and then your kid gets better and you're like, they're saved, and you're thinking you're going to have a whole life with your kid, and then they get leukemia and die. So it was really, really sad. And they found out what happened was an adverse and unexpected integration event. And this was something they had no idea it was even possible, but there was a sequence in the viral vector that was homologous enough to a region in our genome, and so the viral vectors were not designed to integrate. But they did, and they integrated into a control region that then triggered uncontrollable cell growth or leukemia. Now, since then, they have fixed this problem. So since then, they've modified the sequence of the viral vector, so this is no longer possible. But this is an example of how, early clinical trials, with just a limited number of patients are important, rather than rolling this out to lots of people. And it's also important that we really know as much as possible about these viral vectors before we use them in patients. And then in 2007, there was another incidence. Julie Moore died, and this was after an adeno-associated virus. This is the type of viruses that I worked on, and this was to treat rheumatoid arthritis. Now, when this occurred, this again was very sad, and it stopped the trial, and everybody was very concerned there might be some other sort of potential problem. But it turned out that this death was, it was not caused, it was due to unrelated issues, so it was not caused by the gene therapy vector. And so then they were able to restart the trial. So there have been a few setbacks, but we have learned a lot from these, and moving forward, this information has contributed to making gene therapy safer. And again, these trials are in patients that have very severe illnesses, where there's a very significant chance of dying, and very soon. They have fatal diseases, or they have diseases that really significantly impact the quality of their life. Now, delivery. There's some other challenges in addition to the ones that were mentioned before. And these include the size of the gene. Excuse me, I already mentioned the immune response, but the size of the gene is important. If you've got a little itty-bitty capsid, you cannot fit, a ginormous gene inside. So this brings up a few different challenges. You can use different viruses that have a slightly larger carrying capacity. Or you can do some really kind of fun things with genetics, is you can have genes that run in both directions on the DNA. Or you can try to have two viral vectors. Now, everything about these viral vectors is exactly the same, except one is going to carry half the gene and the other carries half the gene, and then when they both get into the same cell, then it recombines into one longer gene. So there's a lot of different strategies for doing this. One of the reasons why adeno-associated virus, which is the one I worked with, has some limited uses because it's a very, very small virus. It has a small capsid, so you can only use the small genes. And some genes that you need to fix in patients are larger, right? So then you might use a bigger virus or a different virus. An advantage of AAV, though, is it is really safe. So all of us are infected with adeno-associated virus all the time, and there's dozens and dozens of subtypes of adeno-associated virus, and none of them are associated with human disease. Also, adeno-associated viruses can't replicate on their own. They have to replicate to have a productive infection in combination with another disease, often adenovirus, but it can be others. So if you can target them, right, if you can target them to the right tissues, so they get where they need to go instead of, all taken up by the liver or what have you. If you can get them to the right place, they are very safe vectors. They just can only carry pretty small genes. Your textbook has a really nice table of some of the common viruses that are used in gene therapy. So I mentioned before you can do ex vivo or in vivo gene therapy. And in vivo gene therapy is exactly what it sounds like. You inject or infuse the virus, directly into the patient. Now, ex vivo gene therapy is when you take something out of the patient. So this could be bone marrow, for example. Or maybe you're doing a vein graft, and you take the vein out of the leg, and then it sits in a bath of saline for a while you open up their chest to put it in. And so while that vein is sitting in just a bath of saline, you could be treating it with a viral vector to maybe make it last longer. So in either situation, you're taking the tissue that needs to be treated out of the patient, you're putting it in a dish of some sort, a container of some sort. You administer the gene therapy vector outside of the patient, and then you put it back into the patient. Now, this is inherently safer because you're not just injecting a whole bunch of virus into the patient, modified or not. You can also use less virus, because if you inject virus into a patient, you have to have enough to get all the way through, all of the patient to the desired target tissue. And remember, most of it is going to get wasted in the liver. So you can use less virus and get it directly to the tissue that you need. Now, obviously, this only works for certain conditions, right? So if you want to treat their bone marrow, this is great. If you want to treat a person's liver or pancreas or brain, you can't take the brain out of the patient, treat it, and put it back in. So this works really well in some situations, but not all. So here we've got a list of many of the common viral vectors that are used for gene therapy. Retroviruses have a pretty good carrying capacity, and they can integrate when you want integration into the host genome is random. There are methods, scientists, people investigating this to make it targeted, because random integration can be a problem. Adenoviruses usually exist for short term, so within weeks or months. Adeno-associated viruses, and there's a lot of different therapies that are using adeno-associated viruses, again, because they're considered very safe. Of course, the limitation is they have a small carrying capacity. Sometimes you look for integration, sometimes you don't. If you don't modify it, it will integrate into a specific place on chromosome 19, but the rate of integration is low. So it says 95% of the time integration will be specific, but that's 95% of when it integrates. A large portion of the time it doesn't integrate, and after a few weeks the expression of your target gene goes down, and some of the time it integrates, and when it does, it usually does on chromosome 19. Herpes simplex viruses, modified, they're in use, and also vaccinia viruses are also being used, or other pox viruses, and they have a very, very large carrying capacity. So what makes a good gene therapy virus vector? A lot of the things that we've talked about before, right? You're going to get it to the target cells and the target cells only. If you want integration, it integrates to the correct place. The correct gene of interest gets transcribed and translated at the appropriate levels, and the final gene product is folded properly and can function properly. And then, of course, it's, safe. A really good essay question might be, what makes a good viral vector for gene therapy or oncolytic therapy, or discuss some of the challenges of creating a good viral vector, and how could those be overcome? So we've talked about the in vivo and the ex vivo, and you've got different options. Again, for in vivo, you can direct it injectly into a site, or you can do an IV. Ex vivo, you take them out, and then you treat them outside the patient. Now, something that is new and hasn't had a lot of success yet, but might in the future, is this idea of using naked DNA. And the big problem with naked DNA is it's less efficient. It's less efficient because of uptake. So you inject just naked DNA into a patient, and it's going to get degraded. So how do you actually get it taken up into the cell? And so they're looking at lots of different mechanisms. Electroporation uses electricity to force it into cells. Pressure-mediated delivery, would be like in Star Trek with a hypospray. Ultrasound, lasers, magnetic fields, all of these things, or they're trying to get the DNA into the cell, but this is actually a really difficult thing because you've got a semi-permeable plasma membrane. And your plasma membrane likes to have tight control over what can get in and what can get out. And just a long piece of foreign DNA is not something that your cell wants to let in, because your cell doesn't know if this is harmful or not. As far as your cell is concerned, this could be, bad pathogen DNA, and it doesn't want any of that getting inside. So the big challenge here is with the uptake. Now, that being said, people are still actively researching this, but to date they've had a lot more success using viruses than they have with naked DNA. All right. quick tie-in. So one thing that this is sort of related to is DNA vaccines. All right, so DNA vaccines are still experimental. We don't have any that work yet. Though of interest, the new coronavirus vaccine that they are testing now is a DNA vaccine. So remember, coronavirus is an RNA virus. But if you, in a lab, in a test tube, you make the equivalent sequence out of DNA, and then the DNA somehow, through one of these methods, gets into the patient's cells. Maybe you give them a hypospray, whatever it is, it gets into their cells. And then that DNA has to traffic to the nucleus, it has to be expressed, it has to make an effective protein. That protein, let's say it's a spike protein, a viral spike protein, that spike protein has to then get secreted. And then your immune system has to recognize this spike protein as foreign and then produce antibodies to it. And if this worked, it would be really, really cool. My hopes are not very high, but I would be very, very happy to be disappointed. It would be great news if this worked. Now, they just started trials. We're still a good year away. If everything goes perfectly, we're still a good year away from having this vaccine rolled out to people en masse. And there's a lot of technical challenges that I hope you understand about this now. But if it did work, it would be very cool, and it might open up a new kind of field of using naked DNA for vaccines and for gene therapy. All right, so this concludes the lecture for the oncolytic viruses and gene therapy. I hope you're all doing well.